last month, which was fantastic to see. And we've been running events as well. Those watches, we had one for Venus on August 27th. There were some breaks in the clouds, and a few people did get to see Venus when it's closest to the Earth in broad daylight, which we reckon perhaps only 1 in a million people has ever done. So congratulations if you were one of those. The big story, though, over the last month was the super blue moon. Now, we get super moons every year, blue moons every 3 years, but to get a super blue moon is extremely rare. It hasn't happened for 14 years, won't happen again for another 14 years. Fortunately, the weather cooperated. We got loads of photographs sent to us. We were all over the radio, as you'll hear on astronomy.ie/audio. And we're going to have a special issue of the magazine, next issue, featuring the best pictures from Ireland. There were loads taken internationally, but we want to record and document Irish astronomy, so don't miss that. The second big event, of course, was the Starbecue. We had brilliant talks from Anthony Lynch of Republic of Astronomy. He takes incredible pictures from Ireland. And our evening class tutor, Dr. John Campbell, who is studying a PhD in physics at the University of Maynooth, but kept it fairly simple with drone astronomy. How can you take pictures in the night sky with drones? Turns out you can. They're very good. And I think it's a future growth area. And then I got my talk about the universe. We had the barbecue running, exhibits galore, and a rocket launch by the Irish Rocketry Society to kick everything off. And then the skies were clear from dusk until dawn, not a cloud in the sky. So we got to see Saturn with its incredible rings, Jupiter and its cloud belts, even the great red spot later on, and the wonders of the universe, star clusters, galaxies, etc. Now, if you missed it, don't worry, there's another one next year. There's one every year for the last 30 odd years. And it's on the 31st of August next year. The date varies a bit because we have to move to avoid the full moon. And it's the last Saturday of August next year. I think you can already book it on astronomy.ie/events if you want to make sure you don't miss your place then. The other thing that's been happening is the comet. And we managed to stay up late at the Starbecue for this new naked eye comet, Comet Nishimura. It's been making the headlines a lot. We couldn't see it with the naked eye. It was barely visible in binoculars. Then conditions in Ireland can be a bit hazy at times. But I got our best photographs of it on Sunday morning, actually, well after the Starbecue. A few hardy souls did stay with us till dawn and got to see the comet that way. And I'm going to show those after the talk if you stay with us. And in keeping with other events coming up, don't forget on the social media, we're telling you to watch out for the BBC's Sky at Night programme about the Very Large Telescope, which is part owned by Ireland in Chile. So that's at 9:30 p.m. on BBC 4 this evening. Watch out for that. So loads happening as always. Do stay with Astronomy On. Join the society if you're not a member already. If you are already a member, get lots of people to join. Encourage them. We're always looking for new members, especially beginners, which is whom the magazine is aimed at. And these talks as well are aimed at beginners. We ask an expert in their field to tell the general public about their area of expertise. So we're delighted that Dr. Sean McMahon, who's a reader in astrobiology at the University of Edinburgh, where he also co-directs the UK Centre for Astrobiology, give a talk on that very subject. His research interests obviously include early life on Earth, fossil microorganisms, and the search for life on Mars and other worlds. He's published about 50 academic papers, including a field guide to finding fossils on Mars and false biosignatures on Mars, anticipating ambiguity, both of which can be read for free online. So tonight we're delighted that he's giving our September public lecture all about the search for extraterrestrial life. Over to you, Sean, and thank you very much indeed.
Thank you, David. Thank you very much for the invitation. It's an honor for me to present to this group. And I will now attempt to share my screen. And perhaps you could just let me know if that's working all right once I've got the slides up.
Yep, you're not full screen yet, but you're full screen now. Brilliant.
Okay, so I am going to talk for probably about 45 minutes or so on the subject of astrobiology and the search for extraterrestrial biosignatures. And if you're not sure what I mean by the word biosignatures, I will explain that in due course. This is my area of work, as David was saying there. And although astrobiology is a very broad subject, my area of specialism within that subject is the search for life in rocks from the early Earth and Mars. So if you have any questions about that at the end, I'm particularly qualified to answer them. So what is astrobiology? Well, there is a sort of NASA definition of the subject, because NASA played an important role in kind of midwifing this subject in the 1990s and early 2000s, and helping to establish its global presence in universities and research institutes. And their definition is: astrobiology is the study of the origins, evolution, distribution, and future of life in the universe. So in other words, it's not just about looking for aliens. It's about trying to understand how biological phenomena, life, fit into our understanding of the universe, stars and planets, and the space environment. Obviously, a big part of that is looking for aliens, but it's not just that. So astrobiology includes fundamental questions about biology. When did life arise on the Earth, and in what kind of environment, and by what kinds of processes? What actually is life? Do we expect life on other worlds to be, well, if it exists at all, do we expect it to be the same as life on Earth or different? How different could it be? And then there are questions related to that about habitability. So what makes an environment, or a planet, if you like, able to sustain life? What are the conditions that must be met for life to take hold and thrive? We'll talk a bit more about that later on. And then once we have some ideas about that, we can use the tools of astronomy and planetary science to look for those conditions and find out whether parts of our solar system are habitable, whether other planetary bodies outside our solar system might be habitable. And that's a first step towards actually finding life. But then, of course, there's the question, well, how do we find life? What are we going to look for? What's the kind of evidence that we can actually measure that might tell us that there's life on another planet, or that there was some time ago in the past? So in this talk, I'm going to talk a little bit about these first two kinds of questions, about the nature of life and what we mean by habitability. And then I'm going to really focus in on what we mean by biosignatures and what kinds of evidence we might be able to find for life, particularly on other planets, but also on the very early Earth, and we're trying to understand how life began. So we might start then by thinking about what we actually mean by this word life. And the fact is, we don't really have a very good scientific definition of life. There's lots of debate actually about whether it's possible to come up with a definition, whether we even need one. But we can point at some of the essential features that all living organisms share, and that we expect anything that we would call life to share, even if it's very different from life on Earth. So those include, for example, encapsulation. So all living things have a kind of boundary around them, whether it's the cell wall or your skin or the bark of a tree. And they have this ability to control the exchange of energy and materials across that boundary in order to maintain a stable internal environment. So for instance, think about your body temperature. So we call that homeostasis. This is fundamental to all organisms, and we can't really imagine some form of life that wasn't able to do that. Another essential feature of life is the ability to acquire energy from the environment and use it to do work, to do the work of living, right? Dividing, repairing cells, making molecular machinery, and so on. So some organisms, like us, for example, we oxidize the sugars in our food. There's a difference in the energy between the reactants of that process and the products of that process, and that energy that's kind of released from the reaction is the energy that we then use to support all the processes of life. Many microorganisms use very different chemical processes, where instead of oxidizing sugars, they're oxidizing hydrogen or minerals or metals, all kinds of things. And of course, there are the plants and algae and types of bacteria that actually use solar energy as their source of energy. But whatever type of energy it is, you can't build something as complex as a living thing and maintain it without absorbing energy from somewhere. All living things also develop according to a kind of genetic program. So in our case, in the case of all life that we know about on Earth, that program is stored in DNA. In the case of some viruses, which are not usually considered, strictly speaking, alive, although there's some debate about that, the genetic code can be stored either in DNA or in RNA. And maybe for life on other planets, it'll be some other molecule that has this long polymer structure in which you can kind of encode instructions that can be read by the machinery of that organism. And then, of course, there's evolution. The passing on of inherited characteristics from generation to generation, which entails adaptation to an environment, the ability to sustain a lineage over time. Life as we know it would be impossible without evolution, although of course you might wonder whether the very first organisms assembled themselves via some other process, since clearly by definition they couldn't have had living ancestors. Then there are features of life that we might think of as not essential, features that we see in life on Earth that could be very different in life on other planets. So one example that's often talked about is the reliance on liquid water, which is essential to all life as we know it. But maybe on other planets, there could be forms of life that don't use water, but have some perhaps other molecule that acts as the solvent. So biochemistry that we know about on the Earth, a lot of the reactions of biochemistry take place in solution, dissolved in water. The way that cell membranes on Earth are stabilized involves critically liquid water. And water is continually being used up and released again by biochemical reactions. So life as we know it is absolutely critically dependent on water, but maybe for aliens, it's something else. Similarly, all life that we know it is based on compounds of carbon. So biomolecules, the things that we're made of, are things like amino acids, proteins, lipids, so fats, carbohydrates or sugars. And all of these compounds have a kind of backbone chain of carbon atoms or rings of carbon atoms. Maybe life on other worlds uses something other than carbon, like perhaps silicon is sometimes talked about. We can speculate about this. Similarly, the other elements that life as we know it is made of, so particularly nitrogen, oxygen, phosphorus, and sulfur, and hydrogen, I might have said, these along with carbon are the elements that constitute all the kind of key molecular machinery of life on Earth. Maybe on other worlds, life doesn't need one of those or two of those or all of those and just builds itself from something completely different. It's possible logically. This little title that you see here, a bacterium that can grow by using arsenic instead of phosphorus. This was a paper that was published in Science a few years ago, which reported the discovery of a bacterium on Earth that supposedly was able to substitute arsenic into its DNA, where all other organisms have phosphorus. Now, it turned out that that was a mistake. There was a problem with their experiment, and this bacterium wasn't actually doing that. But one of the reasons it was very exciting, one of the reasons it was in Science, was that if that had been true, then it would have really expanded our horizons for thinking about what life on other worlds might be like. One more thing that sometimes people talk about as perhaps being different on other worlds is the fact that all organisms on Earth use the left-handed form of amino acids and the right-handed form of sugars. So these are molecules that come in left and right-handed forms, which, just like your left and right hands that you might hold out in front of you to see how this works, you can't fully superimpose them on each other. Your left hand couldn't replace your right hand, it would look wrong. In the same way, they're not, these molecules come in pairs that can't be just superimposed on each other. But maybe life on other planets uses right-handed amino acids or left-handed sugars. We don't know, but there doesn't seem to be any fundamental reason why not. I must say, however, that although we can speculate about these things, there's plenty of water in the universe, and there's also plenty of carbon and nitrogen and oxygen and phosphorus and sulfur. So it's not essential for us to consider radically different forms of life at this stage in our search for extraterrestrials. There's plenty of exciting possibilities for life that's actually quite similar to life as we know it on planetary bodies in our solar system, where we know that there are carbon-based compounds, we know that there is liquid water, and so on. So maybe we want to exhaust looking for these more familiar signs of life before we think about the really radical alternatives. Okay, so where are the aliens? Well, clearly we don't know. Maybe there aren't any. But one thing to think about is that in order for a planet to be able to sustain life, there are two conditions that must be met. In the first place, that planet or that environment, perhaps inside a planet, somewhere in particular on the planet, must be suitable for life to originate there. It's no good having a planet that could theoretically sustain life if it doesn't have the means of actually producing its own form of life. And then again, of course, it's no good having a planet that can generate some form of life but not sustain it. So it's only where these two criteria are met that you might have planets that are inhabited for any length of time. And the reason I'm emphasizing this is that although we talk a lot about habitability in astrobiology, and I'll talk more about that later, we'll need to think not only about whether a planet, has water and carbon and ticks all the boxes to be a habitable environment. We also need to think about whether that planet actually has what it takes to generate life in the first place. And that means we need to understand something about the origin of life. And so you can already see how, even if you just start with that question, is there life elsewhere in the universe? You very quickly start needing to think about the origin of life and how that worked on the Earth and bring in these other areas of science. Now, we don't know a great deal about how life actually began, even on the Earth. We do know that organic molecules, and I'm using the word organic in the technical sense here, where an organic molecule is one that has carbon and hydrogen, doesn't necessarily mean it has a biological origin. We use the word organic not to mean biological, but to mean made of carbon and hydrogen and some of these other elements. Anyway, these molecules are actually very widespread in space. We find them in meteorites. This little pie chart here shows the breakdown of some of the molecules that we find in meteorites. They come from space, things like carboxylic acids, alcohols, amino acids, which are the building blocks of proteins. And we know there are many different possible sources of organic molecules, but we don't really know which ones were most important on the early Earth, or indeed on other planets. We know that life was present on the Earth by at least 3.5 billion years ago. So the conditions for the origin of life had to be around before that. And this picture shows a kind of fossil microbial colony in a 3.5 billion-year-old rock. And we know that the first organisms were microscopic and single celled, as indeed are most of the organisms on the Earth, just in terms of the diversity of life on Earth. And if you look at the way that organisms that we find on the Earth are related to each other and work out their family tree, you quickly realize that the ancestors of all living organisms were quite simple microscopic single-celled organisms. And then there are various things we don't know, which you may come across in your own reading, or you might have heard about. So it's often speculated that these things called hydrothermal vents on the sea floor might have played a role in the origin of life. These are environments where hot fluids discharge from the crust into the oceans, at the base of the oceans. And they immediately react with the seawater and precipitate minerals that form these huge chimneys. These are some submersibles here in this image, if you can see my mouse. These can be as tall as a 6-story building, these mineral chimneys. But more importantly, they're full of little compartments which are chemically out of equilibrium with their surroundings, which is quite a promising thing for getting the chemistry of life kick-started. And we know that organic molecules are generated in these environments. And there's various theories about how the first cells might have come together in this sort of setting. But there's lots of other settings as well that might have played some role in the origin of life. You might have heard about Darwin's idea of the warm little pond. People still talk about the idea that maybe you have water on the surface of the early Earth that's drying out and wetting again, or you have a tidal environment. The thing about evaporation is that it's really good at concentrating organic molecules and bringing them together. We don't know much about the roles of atmospheric chemistry, so sort of nitrogen chemistry in the atmosphere of the early Earth in generating important compounds like RNA maybe. There's speculation about the roles of UV and ionizing radiation on the early Earth, minerals that could have catalyzed the reactions that helped lead to the origin of life. There's this idea that to get out of the chicken and egg problem of which came first, was it a DNA that's made by proteins, or was it the proteins that you need DNA to code for to make in the first place, that maybe instead the first organisms were just made of RNA, which can act both as a set of instructions and can also catalyze reactions. So it kind of fulfills the roles of both DNA and proteins. So these are all some ideas that are discussed a lot, and there's a lot of kind of speculation, controversy, and debate really about what actually was important for leading to the origin of life on Earth. And I won't talk much more about that today. So much for this left circle, the possibility for life to originate in some environment, some planet. Maybe it requires hydrothermal vents, maybe it requires warm little ponds. We don't really know exactly. Let's think about the other circle here. So what does it take for life to persist on a planet? What does it take for some environment to be habitable, or some planet to be habitable? I'm using this word habitability a lot. In astrobiology, we tend to think about habitability as a term of art that has quite a specific meaning for us. And what we mean when we say an environment is habitable is that at least one type of organism that we already know about could live there. So if you have some environment, you want to know if it's habitable. Well, if there's some known organism that could survive in that environment, then we consider that to be a habitable environment. Now, you might well think, well, what about unknown organisms? Couldn't an environment or a planet be habitable to aliens, but not habitable to any life on the Earth or any life that we know about? And of course, that's possible, but we don't know anything about what unknown organisms might need to live. So that's why we just use this restricted sense of the word habitable when we're talking about whether extraterrestrial environments are habitable. So to be habitable, an environment needs carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. All known organisms are absolutely dependent on these elements. There are various other elements in the periodic table that are important for most organisms, or even for all organisms, but don't play quite as essential a role in building the molecules that life is made of. So for an environment to be habitable, it needs these. You can think of them as the most basic nutrients that all organisms need. You won't be surprised to hear that also we consider water to be essential for habitability, given the fundamental role of water in life, which I've already mentioned. Now, we talk about in astrobiology this parameter called the water activity, which is a thing you can measure. There are some materials that have a lot of water in them, but they have a low water activity because that water is chemically bound up and not available for life. So for instance, something like peanut butter, you might have a jar of peanut butter on the shelf. It might have been there for years and it will never have gone moldy because even though there's water in the peanut butter, the activity of that water is too low for any microbes to grow there. So this is a quantitative parameter, and we say that a water activity or the lower limit for the water activity that we know of that life needs is about 0.6, where 1 is just pure water. There needs to be some source of energy to sustain metabolism. And also, there are lots of physical and chemical stresses that can make an environment uninhabitable, even if these other criteria are met. So we think about things like pressure, temperature, UV radiation, ionizing radiation. These things can destroy organisms and make an environment uninhabitable. Also things like pH. Some environments are too acid or too alkaline for life. Some environments are too saline. This picture here is an electron micrograph of a bacterium called Deinococcus radiodurans, sometimes called Conan the bacterium, which is often studied as an example of an extremophile, an organism that's really good at reproducing itself in very extreme conditions. It can withstand radiation doses, so doses of ionizing radiation, of more than 10,000 joules per kilogram of biomass. So for comparison, a human being would be killed by a dose of about 3 joules per kilograms. This thing is really a beast. It could survive in a nuclear reactor. In our solar system, there are a few environments that we think of as marginally habitable or habitable. The surface of Mars is a marginal one. So the surface of Mars today, the pressure is about 6,000ths of the atmospheric pressure on the Earth, which is very low. And the surface temperature averages about -60 degrees C, although it fluctuates a lot. And these two conditions together mean that liquid water is basically never present for very long on the surface of Mars because it sublimates away into a vapor. And without liquid water, as you've seen, an environment can't be habitable. So it's a very dry environment. It's also very oxidizing. The soil chemistry is very oxidizing. So if you think about bleach as an oxidizing agent, this is stuff that's destructive to organic molecules. The surface of Mars is also intensely irradiated by UV and ionizing radiation that tends to, again, destroy organic molecules. So maybe there are organisms that could just about cling on in these conditions, particularly in the thin films of water that might form around salt crystals, things like this. There are various hypotheses about how Mars might just be habitable, but it's probably uninhabitable, at least on average over time. We don't expect to see organisms now at the surface of Mars. 4 billion years ago, it was a different story. And this is why rovers like the Perseverance rover that's on Mars now, and the ExoMars rover, Rosalind Franklin, which should be going to Mars in a few years, this is why they're effectively looking for fossils and not evidence of modern life. They're looking for fossils because 4 billion years ago, we know that there was liquid water, lots of it, on the surface of Mars. We know this because of the minerals that formed, salt deposits, clay minerals. Also from the geomorphology, we can see river valleys that were etched by rivers. So this implies that the temperature and the atmospheric pressure were higher at that time, which then also implies that there would have been less ionizing and UV radiation reaching the ground. Taking all these things together, we think early Mars was actually habitable. This is pretty much a consensus now. And that's why we're looking for biosignatures that formed in that time rather than organisms alive today. It's also possible that the subsurface of Mars could be habitable today. There could be aquifers, bodies of liquid water underground, kept in a liquid state by heat from the planet's interior and protected against radiation from space by the overlying rock. So this is a nice hypothesis. It's supported by the fact that there are environments deep underground on the Earth today, which are quite Mars-like in all these ways, where we know that there are thriving microorganisms. But it's a very difficult hypothesis to test, unfortunately, because we're talking about miles below the surface of Mars. We have no way of getting there. Very difficult to find out if there is, in fact, anything living there with present technology. Another place we talk about as potentially being habitable in our own solar system, or places, are the icy bodies in the outer solar system. So places like Saturn's moon Enceladus, Jupiter's moon Europa. These bodies have subsurface liquid water underneath an ice shell. And the heat that sustains that water in a liquid state comes partly from tidal forces. So the gravitational interactions between these moons and their host planets, the stretching and squashing of these planetary bodies melts what would otherwise be ice, and you have liquid water underneath the ice. And we know that water is there because some of it's actually venting out to space through cracks. And that's what you can see in this picture in the background here. This is an image of Saturn's moon Enceladus. These white plumes are plumes of water escaping to space, and they're actually building the E-ring around Saturn. Now, excitingly, the Cassini spacecraft has flown through these plumes of water and measured their composition. And so we know that that water is quite alkaline, but not necessarily too alkaline for life. We know that it contains organic molecules, so not evidence of life, but a key criterion for habitability, these carbon-bearing molecules. There's also phosphorus in the form of phosphate. There's also molecular hydrogen present in these plumes. Now, the reason that's important is that we know it's produced by chemical reactions between rocks and water, and those reactions could provide energy for microorganisms. So it looks as if the oceans inside Enceladus are really ticking a lot of those boxes to be a habitable environment. And that's one reason why missions are scheduled to go to these icy moons in the 2030s and have a look at these environments and assess their habitability further. We can also talk, of course, about the habitability of exoplanets, planets in other solar systems. And in this context, we talk a lot about the habitable zone. So for an Earth-like planet, the habitable zone is the range of distances from the star in that system in which an Earth-like planet could have liquid water on its surface. And that's a function largely of the temperature. So you might think of it as the Goldilocks zone, where if you're too close to the star, it's too hot. If you're too far away, it's too cold. There's a little bit more complexity to it than that because of feedback processes which help to stabilize the surface temperature of a planet, a bit like a thermostat. And there's an example of one of these processes summarized by this equation here, which involves carbon dioxide. So carbon dioxide, as you all know, is a greenhouse gas. And one of the things it does is it combines with water to form carbonic acid, which reacts with minerals on the surface of the planet. Now, if you took the Earth and warmed the Earth up considerably, which is something that we're now doing, one of the effects of that would be to accelerate this reaction between carbonic acid and rocks on the surface. That pulls down carbon dioxide out of the atmosphere, thus reducing the greenhouse effect. And this therefore dampens the heating effect that you started with. And because of this, the outer edge of the habitable zone, sorry, the inner edge of the habitable zone is a bit further in than it would otherwise be. The other thing that happens is that if you move the Earth too close to the sun, you put so much water vapor into the atmosphere that eventually you just saturate the atmosphere with water and start losing water to space. And then you have a runaway greenhouse situation, because water itself is also a greenhouse gas. At the outer edge, this feedback process, which would otherwise help to keep the Earth warm because the consumption of carbon dioxide is less efficient at lower temperatures. What happens at the outer edge of the habitable zone is that feedback process shuts down because you can no longer build up any more carbon dioxide in the atmosphere simply by shutting down this reaction. And so what happens then is that all the water on the surface of the planet freezes over, and you end up just freezing over the surface of your planet. So that's what we really mean when we talk about the habitable zone for exoplanets. But there's a couple of really important caveats to bear in mind when you read about some planet being in the habitable zone. The first one is that, as we've just seen, the way the habitable zone is calculated assumes that the planet we're talking about is an Earth-like rocky planet with Earth-like atmospheric feedbacks between gases and minerals. If we're talking about a gas giant that maybe doesn't even have a rocky surface, then it's kind of irrelevant that it happens to be in the habitable zone. And I mention this because it's sometimes annoying that you see in the news, new habitable zone planet discovered, or a few years ago, water vapor detected in the atmosphere of a habitable zone planet. And fine, it's not wrong, but it's sort of irrelevant that that planet was in the habitable zone, because a planet that's 8 Earth masses is not going to have a rocky surface. So it's not going to be a habitable planet in the sense that it's going to have water on a rocky surface. It's also the case that for some solar systems, even though the planet might be in the habitable zone by this definition, for certain stars, particularly certain dwarf stars, they're constantly spewing out forms of radiation that would sterilize the surface, even if it might otherwise have liquid water. So the fact it's in the habitable zone then is again sort of irrelevant. And it's equally true also that because of the possibility of life in the subsurface for some planetary bodies, it's sort of moot whether or not you can have liquid water on the surface. Of course, places like Europa and Enceladus, for example, are outside the habitable zone for our solar system. Mars also is just outside, but as we've said, you could still have life on Mars, just not on the surface. It would have to be in the subsurface. So these are some important caveats when you hear about habitable zones. Okay, so now I want to move on from talking about the nature of life and habitability and talk a bit more about biosignatures and the search for life. Now, there are a number of reasons why it's really difficult to find extraterrestrial life. In the first place, we don't know exactly what we're looking for. Secondly, we don't know if what we're looking for even exists. And thirdly, even if it does exist, we don't know what it will look like. So this is a pretty difficult set of situations, set of circumstances. Importantly, we don't know how what we're looking for will be different from what we're not looking for. In other words, we might find things where we're just not sure if they're life or not. We might find forms of non-biological complexity that are not what we're looking for, but we're not sure if they're what we're looking for. And if this sounds like a bit of a tongue twister, the main points will hopefully come across over the following slides. So I'm going to be using this word biosignature a lot. I've probably used it a lot already. This is another one of those terms that has various sort of technical definitions. But basically, if we find life on a distant planet, it's not going to be that we have something we can hold in our hands and say, look, this is an organism. It's going to be that we have some data, we have some information, maybe from a telescope, for example, where that data allows us to infer the presence of life indirectly. So that sort of proxy for life is what we mean when we talk about biosignatures. And one definition that's quite popular is that a biosignature is an object, a substance, or a pattern whose origin specifically requires a biological agent. So you might think of a dinosaur skeleton. A dinosaur skeleton specifically requires a biological process to produce it. It's not something that's going to be produced by any kind of non-biological chemical self-organization. So that's definitely a biosignature. DNA is another thing that we often talk about as a biosignature, but we're not actually strictly sure that it is one, because it might be the case that the kinds of complex carbon chemistry involved in the origin of life are able to produce small quantities of DNA, even in the absence of what we would, strictly speaking, consider proper life. Another feature that's often talked about as a potential biosignature is something called the vegetation red edge. So this is a spectrum. It's essentially a plot that shows the color you can think of this as the surface of the Earth, although this spectrum is actually just measured on a leaf. The key thing to notice here if you look at a leaf or any green photosynthesizing organism, and you look at how it reflects and absorbs light of different wavelengths, these things are really, really good at absorbing the visible part of the spectrum. So between about, let's say, 400 and 700 nanometers. This is the same part of the spectrum that we can see. And the reason they're really good at absorbing that is that they use it for photosynthesis. They tend to reflect a little bit more strongly in the green part of the spectrum, you can see here, which is why these organisms are green, but they're basically really good at absorbing visible light. They're really bad at absorbing light in the infrared, these longer wavelengths here. So that's why you see the reflectance increases at around 700 nanometers, which is the boundary between the visible light here and the infrared light. And so you have this sharp discontinuity, this kind of cliff-like edge feature. You see this when you take the spectrum of a leaf. You can also see it if you point a spectrometer at the Earth from space. So even a distant telescope pointed towards the Earth, measuring the spectrum of the Earth, could see this feature. So is it a biosignature? Well, that depends on whether there's some non-biological process that could produce the same type of edge-like feature in the spectrum of the Earth. And I'll suggest later on in this talk that there might be. You might think all of these examples of biosignatures are kind of hopelessly parochial because they all just focus on things that life as we know it can do and don't take into account the possibility that life on other planets might be doing something very different. And so there are efforts to try to come up with biosignatures that we can measure that are a bit more agnostic with respect to the form of life that's actually producing them. So one thought is, never mind what your molecules actually are, if they're DNA or proteins or something else, let's just look at how complex they are and measure that. And any form of life, even if it doesn't use DNA, even if it's not made of carbon or whatever, its molecules will still have to be really complex. And there are various ways of measuring that. And this is a plot from a paper that attempts to do that, to make this measurement for various different substances. And on the y-axis of this plot is something called the molecular assembly index, which is a measure of how complicated these substances are. It's based on mass spectrometry, but that's not important. And that point that I've highlighted there is E. coli. So this is a type of bacterium, and you can see it has a really high value. It's made of really complicated stuff. Whereas all these low values down here, most of them are things like minerals or simple fluids that are not biological in origin. Interestingly, the highest value here comes from beer. Beer is among the most complex substances known to man whose complexity has actually been measured in this way. So beer is a biosignature. Okay, so as I've already hinted, looking for life is difficult. There are lots of things that can go wrong. And here's a helpful way of thinking about that. If there is a biosignature in the environment, then we have a yes answer to this question. Is a biosignature actually present? Can we detect it? Yes, then we have a true positive. Great, we found life. But if there's a biosignature present and we can't detect it, then we call that a false negative. We have a negative result, but it's wrong because there is life actually there, but we're not seeing it. Similarly, we can get false positives, where we think we've detected a biosignature, but life isn't actually present in that environment. We've made some kind of mistake. And of course, we can have a true negative where we don't find life and there actually isn't life there to be found. So what we want to do in our approach is maximize the true positives and the true negatives and minimize the false positives and the false negatives. Now, here are some examples of reasons why we might come across false negatives. So one reason is that we're often looking for biosignatures in environments that are quite extreme. So even if life is there, anything it's producing that we could measure is quite quickly destroyed. And an example of that would be the surface of Mars, where the ionizing radiation is so intense that it would destroy 99.9% of large organic molecules in a geologically short time span in the upper part of the soil. This is a plot showing the measured radiation dose on the surface of Mars. It's always pretty high, and sometimes it spikes extremely high. This is why the rovers being sent to Mars but this is partly why they're looking for molecules in ancient rocks where they can drill out a sample from inside the rock and not rely on material at the surface that's been exposed to radiation. So this is one source of false negatives, the destruction of biosignatures by extreme conditions. Another thing is that sometimes the things you want to see are just really, really faint. So I mentioned before the vegetation red edge. Actually, it's a really faint, the figure I showed you before was like an ideal case where you just measure a leaf. In reality, the spectrum of the Earth has all these other features in it due to other things present on the Earth. The red edge, which is this little slope here, is a really small feature. Of course, it comes and goes depending on which side of the Earth you're looking at, because you might be looking at the side with more vegetation on it or the side with less vegetation on it. So that's an extra clue that this is a biosignature. But the same, of course, might be true of minerals that have the same type of spectral shape, and that they might be more concentrated in certain areas than others. And so altogether, these types of reflectance features in the spectrum of a planet might just be so faint that you can't really see them, especially when we're looking at exoplanets where the quality of the spectra that we can observe is going to be much worse than these spectra here. By the way, these spectra were obtained from looking at the light reflected from the surface of the Earth onto the moon and collected from the moon. Another example of a false negative would be the sort of problem that comes about from just using a non-ideal measurement technique. So an example of this would be the Viking missions to Mars, which in the 1970s actually measured the presence of organic molecules on the surface, but nobody realized that organic molecules had been detected. That's because the means of measuring them was to take a sample of the soil and burn it in an oven and measure the breakdown of organic compounds. And what happened when they did this was that they measured compounds that had things like chlorine in them and fluorine. Those compounds were attributed to the presence of things like bleach that had been used to clean the spacecraft. They were present at quite low levels, and no other organic compounds were present. It turns out now we understand that probably there were organic compounds in the soil of Mars, but there were also perchlorate compounds, a type of salt. And in the oven of the instrument that was used, the salts and the organic compounds reacted together to produce these things that looked like bleach, like cleaning agents, because that's what perchlorates and organic compounds do at really high temperatures. So this was understood much later, and we now know from other missions that there are small amounts of organic compounds on the surface of Mars. Again, this is not evidence of life, although it is evidence of habitability. These compounds are delivered by meteorites, as we've already seen. But we had a false negative in our search for organic compounds back in the 1970s. There are even environments on the Earth where life is present at such low concentrations that using all the tools of modern Mars rovers and so on, we would really struggle to detect it. And there was a paper describing this situation published earlier this last year. The image on the left here shows a desert environment from Chile on the Earth. The image on the right shows a very similar environment on Mars. In the environment on the left, there were organisms present, but they were so rare that they wouldn't be detectable to rovers. So we would get a false negative result if we tried to look for these organisms using those methods. Then, of course, you have the false positives. And unfortunately, the history of astrobiology is littered with examples of false positive detections where we thought we'd found life and actually we hadn't. And I'm going to go through some classic examples now. So you may have heard of Percival Lowell, a Harvard astronomer who at the beginning of the 20th century thought he could detect canals all over the surface of Mars with his telescope in Arizona. And he thought that these had been built by an alien civilization on a drying world to help them move liquid water around and irrigate their cities and so on. He had this whole elaborate theory. Some people never really took this seriously, but some people did. One of those who took the trouble to respond to this claim about life on Mars was Alfred Russel Wallace, who was important as a biologist and evolutionary theorist in the 19th and early 20th centuries. Alfred Russel Wallace pointed out that the idea that these were canals didn't make sense because even from the data available then at the turn of the century, he thought you could conclude that Mars simply wasn't habitable. There couldn't be canals because there couldn't be liquid water, and higher forms of life couldn't exist on the planet. So this is a nice example of early astrobiological thinking. And it furnishes us with a nice lesson, which is that it can't be a biosignature if the environment is not habitable. And of course, what happened was those early observations that seemed to suggest the presence of canals were kind of progressively debunked over time as telescopes got better. This just shows a sequence of images taken over time. The features that seemed to correspond to canals kind of dissolved as we got better and better images. And finally, Carl Sagan and others in the 1970s, showed that although a small number of those canals might correspond to real geological systems, most of them didn't. They were just some kind of optical illusion, basically, arising from what goes on between the telescope and the human mind and the eye. So these were always very faint, kind of tenuous observations that were just over-interpreted. So that's the lesson here, I think. Another example also happens to feature a Harvard astronomer by coincidence, would be the claimed detection of life on Mars in the middle of the 20th century. William Sinton, who was an astronomer, pointed his telescope at Mars and thought he could see, this was observations taken from the Earth, features in the spectrum of Mars that he thought corresponded to large organic molecules at around sort of 3 microns. And he noticed that these features were more pronounced when the darker regions of Mars were pointing towards Earth than otherwise. And at this time, it was widely believed that the darker regions of Mars corresponded to the presence of some sort of simple vegetation. We now understand it's actually just rocks. This was important in the history of astrobiology. These claims were being made in the late 1950s. NASA established their exobiology program in 1960. A key person in the early NASA exobiology program was Josh Lederberg, a Nobel Prize-winning molecular biologist, who was actually convinced that this was good evidence of life on Mars. And this is just a quote from a paper indicating that. Now, it turned out the real explanation for these features in the spectrum of Mars actually has nothing to do with Mars. These features are contributed by heavy water molecules in the atmosphere of the Earth that happen to be along the line of sight when the telescope was pointed towards Mars. The observations when Mars was pointing the darker side towards Earth happened to be made when it was also a slightly cloudier night, unfortunately. So the lesson here is beware of unconceived alternative explanations for your observation that might seem to point towards life. And of course, beware of terrestrial contamination in your signal. Another classic example in a similar region of the spectrum, the great cosmologist, astronomer Fred Hoyle claimed that there were bacteria in space because in interstellar dust clouds, he was seeing spectroscopic features shown by the dots here, very similar to what you would measure if you pointed your spectrometer at bacterium, shown by the line here. Now, of course, the reason for that, these are real features of the interstellar dust, but they're explained by organic matter. In particular, long chains of organic carbon in certain compounds. They're not explained, that they're not bacteria, they're just organic molecules. An important point here is that the building blocks of life, which may be around in the universe, maybe were around on early Mars as well, can strongly resemble life itself. Another classic cautionary tale, the Martian meteorite ALH 84001. You may remember, I certainly remember, Bill Clinton on the lawn of the White House announcing that NASA may have discovered life from Mars. This was because in the 1990s, features in this meteorite were interpreted as tiny little fossil bacteria, minerals that were supposed to have been produced by bacterial metabolic processes, tiny magnetite crystals, a form of iron oxides produced by some bacteria on the Earth, organic compounds again. We now know that all of these features of that meteorite have non-biological explanations that actually explain them better. And there's work about this still being published today, trying to understand exactly how these things ended up in this meteorite. But basically, nobody really takes this seriously anymore as good evidence for life on Mars. So the lesson here is that you can have multiple lines of evidence that all seem to be consistent with life. But if all of those lines of evidence are individually weak, it can still end up that your case for life gets debunked. Controversies like this continue to happen all the time. Very recently, just in 2016, the paper was published in a journal, Nature Communications, arguing that these structures seen on the far left of the slide here, observed on the surface of Mars by the Spirit rover, could be equivalent to these structures in these two panels on the right, which are found around hot springs on Earth, where, according to this paper, these structures that we find on Earth are made by bacteria, which organize silica that's precipitating out of these hot springs into these elaborate branching structures. Now, the problem with that theory is we know that very similar structures can form without bacteria being present or involved. An example is this image over on the right here. If you just take a can of spray paint and spray it onto a surface, the way that the particles interact and build up on the surface will generate little columns and domes and things like that. So that's probably what's really going on around hot springs, both on Earth and possibly also on early Mars. The same thing happens on the floor of car factories, by the way, where the spray paint that's directed towards the cars builds up on the floor. It makes these elaborate branching structures. So it just seems to be something that happens where you have small particles gluing themselves onto a surface, nothing to do with life. You may have followed the controversy around Oumuamua. This was an interstellar visitor in the form of a rather unusual object that entered our solar system, did a sort of slingshot around the sun, then went back into space. Its trajectory was rather peculiar, probably because it was outgassing hydrogen. It's all been more or less explained now. It also had a very strange shape and very strange surface reflectivity properties. These have all been explained now, but that didn't stop. And yet another Harvard astronomer, I don't know why it's always Harvard astronomers, but from claiming that this was in fact an extraterrestrial spaceship, a light sail, etc. There's also been controversy about phosphine pH3, which was observed in the clouds of Venus and compared with phosphine on Earth, which is a compound produced abundantly by bacteria. It's quite hard to explain why there would be high concentrations of phosphine on Venus, whether or not there are bacteria, because it would be very difficult for anything like a bacterium that we know of to survive in that environment because it's extremely acidic and has very, very low water activity. And it's also unclear whether the detection is real. That's still controversial. But anyway, even if it is real, it's not a biosignature, certainly not by any kind of analogy with life on Earth, if the environment is totally uninhabitable to any organism that we know of that could produce that signature. So that's my take on this. So the score so far is pretty bad. Lots of false positives, possibly a false negative or two along the way. We're not really sure. No real evidence for life. Along the way, as we've been through these kind of cautionary tales, I've tried to highlight some of the things we can learn from them. Obviously, it's okay for science to make mistakes as long as we learn from those mistakes. But we might want to think about how to avoid these kinds of mistakes in the future. One suggestion is to avoid false biosignatures, false positives, by studying the non-biological processes that might generate lifelike phenomena, which could lead us astray. And then we can learn how to discriminate true from false biosignatures. So oddly enough, this means astrobiologists have to be specialists in non-living features of extraterrestrial environments, the chemistry, the physics, the geology, as well as the types of biology that might possibly survive there. The figures on this slide show an example of something that used to be thought to be a fossil, this dendritic pattern that we find on certain rocks, where now we have a really good understanding of the physics and chemistry that produce this pattern. We can simulate it in a computer. That's what this thing on the right is. We know that life isn't involved. This is the level of understanding we need to eliminate false positives in our search for biosignatures. Unfortunately, nature is complicated and does all sorts of interesting, complex things that aren't alive, particularly through what we call self-organization. So in environments where you have some kind of energetic imbalance and where you have feedback processes, matter can move around and organize itself into interesting structures that can look misleadingly lifelike. So that can range from things like convection cells in the mantle of a planet, or in your soup on the hob for that matter, where you might see hexagonal patterns forming, to a whole range of different patterns that we see on the surface of Mars, which we know are self-organized, like these spiral features that we see in some lava fields on the surface of Mars, where we understand that these spirals that look a bit like seashells are actually a type of cooling fracture that can appear in lava when it cools down. And all these other patterns are also kind of large-scale, self-organized patterns that we see in orbital imagery of the Martian surface. In rocks on Earth, we find all kinds of interesting self-organized patterns that can look a bit like fossils or a bit like the products of microbial growth, but we know that they're actually not. They're just non-biological self-organization processes. Here are some examples from various rocks on Earth and things we can grow in the laboratory. And here's a set of images of things that we can either find in rocks or grow in the lab from realistic environmental conditions that we simulate, where all of these things look a bit like fossils that we might hope to find on Mars. So some of them are made of organic molecules. They often have complex shapes that look like dividing cells or filamentous sort of fungal growths, this kind of thing. All of these things are non-biological. This is from the paper that David mentioned at the beginning of my talk, False Biosignatures on Mars, which outlines the scale of this problem. So I think it's by studying these kinds of systems that we put ourselves in the best position not to be fooled when possible evidence for life on Mars comes in. Just to give one more example of a debate that I've been a bit involved in, there was a paper published in Nature a few years ago that described tiny little mineral tubes of iron oxide that were found in a hydrothermal vent deposit about 4 billion years old from the Earth. And the paper compared these tubes to similar structures made by bacteria on the Earth and argued that these were fossils that should be primary targets in our search for extraterrestrial life. And they were thinking particularly of Mars there, where we know that materials of a similar composition to this, that is to say, lots of iron, lots of silica, hydrothermal mineral deposits, may exist or do exist. So they thought there's no non-biological process that can produce that structure. One of the things I've done in my work is to show that there is a non-biological process that can produce exactly that type of structure with the same composition and the same type of rocks. So I'm not saying that it's definitely not a fossil. I'm just saying it's not a very reliable guide to, it's not a very definitive biosignature, let's put it that way. So the image on the right here is the thing I made in the lab, and the one on the left is the one from the paper that they found in an ancient rock and claimed was a biosignature. And this process, by the way, that produces these is absolutely not biological at all. Just another example is the possibility of finding oxygen in the atmosphere of an exoplanet. This is often talked about as a kind of ideal biosignature that you might look for, but we know that there are non-biological processes that can produce a buildup of oxygen in an exoplanet's atmosphere, in particular via the breakdown of water molecules by UV light. And so there's lots of discussion now about how we could be sure that the oxygen we were looking at comes from life rather than this other process. And I won't go into any more detail about that because I know I'm going a bit over time here. And just to say one more thing, the same is true about the vegetation red edge. So this is something we're working on now here in Edinburgh. This spectrum here, that you can see this edge feature at 700, 750 nanometers. This is a spectrum we take from a leaf. And this blue one is a mineral that we found that has a very similar feature in its spectrum. And so if you find a similar feature on the surface of a planet, we're not necessarily going to know whether it's some form of vegetation there or whether it's just some mineral like this. So that's something we're currently working on. Okay, so to summarize, and I'll take questions after this, life detection in astrobiology is really challenging. It's often difficult to tell the difference between evidence of biology and evidence of maybe non-biological processes that we just don't understand very well. This can lead to both false positives and also potentially false negatives in the search for life. And so astrobiologists have to understand the non-biological context in which they're looking for life. That means they have to learn about astronomy and geochemistry and geophysics and so on, planetary science. Only then are they going to be able to tell the difference between the signal of life and the kind of non-biological baseline against which we're hoping to recognize that signal. And that's the end of the formal part of my presentation. So I'm going to stop sharing my screen now. And I'm very happy to stick around for any questions that you might have. And sorry for going on a bit there.
No problem. Thank you very much indeed, Sean. We're going to take questions now if you want to ask them in the chat box, or just unmute your microphone and shout them out. But I want to tell you about the eclipse that's coming next month, which you can see from Ireland, and show you that picture of the comet that we took at Starbecue from Ireland on Sunday morning, and a couple of other interesting things like that. It'll only be about 5 minutes, so do stay tuned after the Q&A. So if you have any questions, put them into chat or unmute your microphone now and shout them out. I'm very shy this evening. Let me start the ball rolling. Just ask your own professional opinion, Sean, and that's, when do you think we will find evidence for even simple microbial extraterrestrial life?
That's a really good question. I think the soonest it's realistic that it could happen is probably, and this is being quite optimistic, but I would say the 2030s. So there's a number of things happening in the 2030s that we can be quite excited about. One of them is that we should have samples returned to Earth from Mars by a shared effort between NASA and the European Space Agency, returning samples collected by the Perseverance rover that's finding them now. It's quite possible that when we bring to bear on those samples the might of Earth's analytical laboratories, we'll find things that are just undisputable evidence for life. Also, beginning in the 2030s, we expect the next generation of space telescopes to come online. That's a very optimistic timeline, but it could happen. There's various mission concepts that are being discussed, which would have the resolution and the signal to noise quality to be able to take a direct image of an exoplanet, where we block out the light from the star and we just get a single pixel, but it's the color of that pixel, the spectrum, that could contain indicators of life, like a particular combination of gases in the atmosphere, plus some maybe color features that look like pigments, something like that. So that's a very optimistic answer. I mean, of course, if you want to be pessimistic, we may never find evidence of life. It's quite possible. So I hope that answers the question. And I see that there's a good one here in the chat.
Yeah. Do you want me to read it to you, or you're happy to read it out yourself?
I can read that out. So Doug asks, is there any biosignature that would be definitive evidence for life? Yeah, there's a few things we can imagine that could be. I mean, we could find, a really complex organism fossilized on Mars. It's possible, where there's just no doubt that it's not the product of some non-biological process. We could find living organisms in the fairly near surface of Mars if they're extremophiles that are just a bit more extremophile than the extremophiles that we know on the Earth. It's possible. There's speculation about that. If you have living organisms and you can grow them, then you don't need to worry about these indirect proxies. You found the thing. If we're talking about exoplanets, I think it's quite unlikely that we'll get something really definitive, but it could be that we have so many different lines of evidence where all the lines of evidence are really strong, like we have oxygen and we have ozone and we have, let's say, a lack of the associated gases that might make you think that the oxygen was non-biological. And maybe we also have several different things that look like pigments. That could be maybe not completely definitive, but pushing in the right direction. There's also, of course, the possibility that we'll discover extraterrestrial civilizations where we're able to decode some sort of message or see signs of their technology. I mean, I think that's quite far-fetched, but I don't want to say it's impossible. I think we should be open-minded about these things. So I hope that answers your question, Doug. Jerry saying, at the end of the day, what's the real odds of finding life in our lifetime? Well, it's difficult to give a probability because it depends on your prior probability for life existing in the first place. What I will say is that if it does exist, I think the odds are actually relatively good, given the way we're going about things, that we will find something in the next, let's say, 20, 30 years. If we don't find any sign of life on Mars in the next 20, 30 years, I think it might be time to face the possibility that Mars just never was inhabited. Similarly for the outer solar system, we're going to get some good data on what's actually in those oceans. And I think if we don't find good signs of life, then we should probably start to entertain the hypothesis that there actually isn't life there.
Could I interject there, Sean?
Yeah, sure, of course.
The chance that, the way life, I think we all probably accept Miller's experiment, etc., that really simple microbial life must have got started, but it was a long time before multicellular life, and some people think that was just a fluke. What's your own professional opinion? Do you think life will start multicellular life will start everywhere, or are you one of those that think it was extremely unlikely and we're probably the only ones?
The way I think about it is that there are kind of a series of, we can call them thresholds or sort of transformative events in the history of life, each one of which might be very improbable. The first of which is the origin of life itself. And yes, it's quite possible that very few planets have an origin of life. And then to get from simple microbial life to anything else is also very improbable. So you end up multiplying these probabilities together and getting a number that's effectively going to be zero, even when you've multiplied across all of these different planets.
And
I
you're
guess
the co-director of the UK Centre for Astrobiology. So
Yeah.
to put you on the spot, what do you,
No,
what do you personally think?
that's fine. So personally, I suspect that what we might call simple life is relatively common in the universe. Other people, it's a matter of intuition, I would say, because the evidence is not really compelling at this point. But I would expect what you might call complex life, multicellular life, to be very rare. So there are some reasons for thinking that, which I'll give you now. So one reason for thinking that is that relatively, let's say, large complex life appeared quite late on the Earth in history. So quite soon after the Earth became habitable, maybe about 4 billion years ago, there was microbial life. The fact that it happened early might lead you to suspect that maybe it didn't require some fluke event, but was actually something that was more or less likely to happen given the right conditions. People don't all agree about that, but you can take that view. By contrast, it took more than 2 billion years after that before we had more complex forms of life appearing. And all of those, what we might call more complex forms of life, have a common ancestor, which means in evolutionary terms, it's something that only happened once. It didn't happen for 2 billion years. And then when it did happen, it only happened once. So that doesn't look like an event that's going to happen a lot. So that's one perspective. On the other hand, there's a hell of a lot of planets. So who really knows?
Yeah, exactly. It's conjecture. I won't hog the questions. A few more. We might rush through the next couple of minutes if we can.
All right, I'll speed up a bit. How important is gravity as a habitability parameter? Actually, not very. And neither is pressure, which of course is related to gravity. The pressures you have to get to before organisms can't survive anymore are so high that you would never find those pressures anywhere on the Earth, except where the temperatures were also way above the habitable threshold. So pressure isn't really that much of a constraint, neither is gravity, on the shape of that habitability envelope. Is the solar system the realistic limit to find reliable evidence of life today? I think probably yes for our lifetimes, unless you think it's likely that we'll find evidence of extraterrestrial civilizations, where, who knows, the evidence could be actually really compelling, but maybe not very likely. What about sending an extremophile from Earth to Mars to see if it would survive? One could do the experiment. I think a better approach is probably just to characterize that environment with lots of different instruments and reproduce it on the Earth. The last thing we would want to do is contaminate the surface of Mars with organisms from the Earth and then no longer be able to tell the difference between indigenous forms of life and things that we've just accidentally put there. And there are various regulations around this at NASA. NASA has a planetary protection officer whose job is to stop Earth contaminating Mars or vice versa. Has the long list of false positive announcements of detection of extraterrestrial life affected the research efforts in this field? I think it has, but I think it's affected the field in a positive way by teaching us how to avoid making these kinds of mistakes and encouraging the kind of research that's needed to avoid them. And also, something that's happening a lot just in the last couple of years is we're getting smarter about how we communicate with each other and with the general public. And there's a lot of discussion now about when we found something that we think might be a biosignature, how do we communicate that in a way that the tabloid press don't just run away with and run this headline that we found life, we found evidence of life, etc. So it's partly kind of a PR issue that we have. Not to get people's hopes up, only for them to be continually disappointed. I hope that's a fair answer to that question. What it's not done is made everybody just really pessimistic. There's still lots of excitement with all these new missions that we're getting closer than ever to finding something if it's there. It's not put people off. I hope I haven't put you off either, by the way.
And that's all the questions? Great.
Okay, if you think of anything afterwards, you're very welcome to pop me an email. You can find my email address online, but I'm also going to put it in the chat here.
Great. Well, we'll leave. Oh, there it is. We'll leave the chat there, and we're going to tell you about the eclipse and the comet just in a second. But let me, of course, thank Dr. Sean McMahon for zooming in to us this evening. I know that he has a very important date with some energy. And even the co-directors of UK Astrobiology Institutes need sustenance. So enjoy your evening, Sean. Thank you very much for joining us. Very thought-provoking talk, as always, is life elsewhere. So
thanks very much. Thanks for your comments. Thanks for having me along. I'm sorry not to stick around for the announcements, but I am very hungry. So I'm going to leave you now. Bye for now, everyone. Bye.
No problem at all. So let me tell you briefly about what's happening in the sky. I'm actually going to leave the audio off because I'm going to share the screen in a moment to show you those pictures, and I might sometimes have problems with my bandwidth, so no sense seeing another picture of me when you want to see the comet. Everything I'm going to briefly mention is in the magazine, which you can buy on the website or hopefully get it already in the post. First is the International Space Station in evening skies starting this Friday. We'll be issuing predictions on all of our social media every afternoon for that night, and if it passes near a planet, the moon, or in front of anything, we'll have the exact place you need to be in Ireland to see that. So there are apps that give you the positions, but follow Astronomy On to get the Irish ones, the best Irish ones, we think. You'll also get Aurora alerts as well. Planets on view? Well, Venus is in morning skies, if you like getting up at the crack of dawn, and we did see it from the end of the Starbecue on Sunday morning. Jupiter and Saturn are the best evening planets. Saturn's up first, about the brightness of one of the brightest stars in the sky. You could see it at Starbecue quite plain with the naked eye, incredibly in the telescopes. And then well to its left, but much brighter, is Jupiter. When it rises around 10 o'clock or so, just before 10, it's the brightest thing in the sky. You can't miss it. Full guide in the magazine. On to other things to see. Neptune is near a star tonight. We managed to show people the planet Neptune, quite faint, even difficult with binoculars, in the telescopes at Starbecue. And as they saw, it was near a star. And tonight is the night it's closest to that star. It's a fifth magnitude star, technically visible to naked eye. Check out the magazine. I'll tell you which one it is and how to find it. You might be able to see Neptune in your own binoculars, even if you don't have a telescope. That's an extremely rare sight. We reckon less than 1 in a million has ever seen Neptune. Then other big events you'll see with the naked eye are the planet Jupiter, as I mentioned. And if you turn your binoculars on, or even a small telescope, and you saw this at the Starbecue, it's very close to a star, about the brightness of its own moons. So sometimes it looks like it's 5 moons. And I've been watching this every night for the last couple of weeks, and it's going to swing back past that star closest to it again on September 17th. Details in the magazine. Now, looking ahead to the end of the month, Mercury is making its best morning show of the year. We're pretty sure less than 1%, well less than 1% of Earth's population has ever seen Mercury. And September the 22nd, give or take a week either side of that, with a few days around September the 22nd, Mercury's at its highest in Irish morning skies. There's a full guide to that in the magazine. Don't miss that if you don't mind getting up early. Of course, it's still summer, despite what everybody's trying to tell you in Ireland and England. The rest of the world, 99% of the world, knows that the seasons are defined by the equinoxes and the solstices. And until we have the equinox on the 23rd of September, it is still summer in the Northern Hemisphere. We're not going to go by what the travel agents say. We're not going to go by what the meteorologists say, because they're based on a human-made calendar, which has nothing to do with the seasons at all. So you'll find the correct definition of summer in Astronomy Ireland magazine. Then on September the 24th, Saturn gets quite close to a star. They all want to be looked at in telescopes to see these stars looking like extra moons this month for some reason. Details in the magazine again. And then, of course, one of the big events at the end of the month is on the 26th of September, Tuesday night. Saturn will get very close to the moon, and the moon's nearly full. 91% of its diameter is sunlit, but it gets about 5 moon diameters away from Saturn. So the full diagram, that in the magazine. You should see it with naked eye, and incredible in binoculars. And then on the 29th of September, we've got our last supermoon of the year. It's also the harvest moon. Friday night, it'll look just as good the night before and the night after. I'm sure you're going to hear all about that on TV and radio as well. And then just briefly, in the next month, we're writing this at the moment for the October magazine, Sky Diary. On October 1st, Jupiter will be very close to the moon. That's going to be really spectacular. One for your binoculars will be later that night. Sorry, the next night. The Pleiades star cluster will be near the moon. And just on the morning of our next lecture, Venus will be as closest to the star Regulus, the brightest star Leo. And then I've promised to tell you a little bit about the eclipse. Well, that's on October the 28th. It's a partial lunar eclipse. The bottom bit of the moon will be eclipsed. The rest won't be. Of course, the eclipse has to happen at full moon. It'll be rising reasonably high in the east, as seen from Ireland. It's a Saturday evening, so nice social occasion. You don't have to get up at the crack of dawn to see something for once. We're writing a full guide to viewing that eclipse in the October issue of the magazine. That'll be out in about end of next week, I think it'll be. So do watch out for that. Circle your calendar, Saturday, October the 28th. Don't make any other plans. Let's hope for clear skies. All these details and more in the magazine. They'll also be reminded to you in the weekly members email, so do join the society if you're not already a member. So I promised you those comet photos. Well, actually, I managed to photo a few things at the Starbecue. Not all of them, of course, there are too many things to see. Let's just see now if I'm load this one up. There we go, give me a second. Hope he's not sharing it and I haven't spoiled the view. Now, can somebody tell me if there's a little blip in the middle of the field of view there?
Yep, you can see it there.
Right. That's the planet Jupiter, as seen through one of the telescopes. You can actually zoom in on this and get some detail, which I hope works on the next picture. There we go. Can you see that one okay?
Yeah.
I think I'm getting the hang of this. So the pictures never do Jupiter justice. I always think the live view in the telescope is better than you can photograph. But you can just about see a small red blip, slightly red, I'd say, on the bottom of the lower. So there's two main cloud belts on Jupiter, like parallel lines, and the lower edge of the bottom one is the great red spot. It just crossed the center of Jupiter. It would have been a bit better earlier on, but people were looking through the telescope instead of me trying to take pictures. You can take lots of pictures, stack them, and get much better detail than this. But at least we managed to record the great red spot, and picture that goes into the magazine, you'll get a chance to process them. These pictures have been unprocessed. I mentioned Jupiter has an extra moon. Well, here's the picture we took at the Starbecue. Jupiter's burnt out, overexposed in the middle. And then we've got three of the moons, and the fourth one up here in the top right is not a moon. It's that star, Sigma, in Aries, that Jupiter's been passing close to, and actually loops around it over the next few days. So it'll stay in the field of view of this particular telescope for a couple of weeks, actually. So watch out for that, even in your binoculars. That was Jupiter we saw. Then we saw Saturn. There's a picture. We actually zoomed in a bit on Saturn, and then I've magnified that a bit more. Again, it doesn't do Saturn justice, what it looked like with the naked eye, but you can see the rings going in front of the planet, how it's tilted. And in the actual telescopes, you could see at least four of the moons. Titan and Rhea were very easy. And we had a map showing which one is which by the way, is still on astronomy.ie/SBQ2. If you want to have a look at the diagram. And then there were a few deep sky objects we had to look at, keeping the best to last. This was the Ring Nebula. Zoomed in a bit, you can actually see the central star there, which is a star that's died and blown off its outer layers, like the sun will in 5 billion years. That was a live picture taken at the Starbecue through one of the telescopes. Another star that died, this is the Dumbbell Nebula a lot of people saw through the telescopes. And they were just some of the objects I photographed. Later on, when most people had gone home, the Orion Nebula rose, got this quick exposure of it. It's a single picture, unprocessed, but it shows just how beautiful that looks. But then the comet rose, and its tail was so long. These pictures are unprocessed. We've got lots of pictures that can be stacked together and processed to bring out much more detail than this. But I thought I'd just get the raw ones up. You can see how long the tail was. This field of view, I think, is about 1 degree long. People are seeing 7-degree tails with less zoomed in lenses. And here's the comet, I think, showing the tail quite well, at least on my screen. Close up, you can see there is structure in the head, and that will come out better when we process the raw pictures. But you can certainly see how bright the head of this comet is. And I could see that in binoculars quite clearly, even when it was low down just above the trees. And then, unfortunately, the twilight rose. So what are the prospects for viewing the comet for the next few days? Not great. It's getting lower. The twilight was brightening up this picture, as you can see. That's going to get worse. The comet is closest to the sun on September 17th, just a few days away now. It could be boiled away and vanish, not be back for over 400 years. Or it could come out the other side and be visible. I think in the southern hemisphere is going to be best when it comes out the other side. We won't see it, but we'll have great pictures of it in the magazine in the next issue. And that's Comet Nishimura, only found in August, which is great advantage for our little Starbecue because it got us on radio a few times as well. The last thing we actually did, so I share this with you, is we saw the moon. And so we took a few pictures of that. And the moon is so huge, the telescope so powerful, you can really zoom in. And in fact, then you can move around the moon to get a close-up look at all those lovely craters. That one up there in the top left edge, you could see the shadow of the crater wall and all the jagged peak shadows. It looked even better with your eyes. As I said, it's hard to get a single picture that will do it justice. And look at that big giant crater with a little crater on its rim. And in the middle of that one, you've got several mountains. The moon's always a fascinating object. We'll have moon watches. Usually best to see the moon in the spring, and we'll have watches then. So that's the pictures of the moon. I'm just going to close by telling you that Sky at Night's on tonight, 9:30 BBC4, about the very large telescope that Ireland has a share in. October the 3rd, our evening classes are beginning. Please tell all your friends. You can enroll online, astronomy.ie, and our next lecture is on Monday, the 9th of October. Remember, they're usually on the second Monday of the month. Robots in Space by Dr. Elizabeth Pearson, who's written a book on this of the same name. And it's not so much robots like the classic science fiction ones. It's the robot spacecraft that we've built that can maneuver themselves around planets on their own with their own clever software. She's also the news editor for the BBC Sky at Night magazine. So Dr. Elizabeth Pearson knows her stuff. Should be a fascinating talk. You can get tickets for it now on astronomy.ie. Just go to the event section and we'll see you all there in 4 weeks' time on Monday, October the 9th. But don't forget to follow us on Facebook, Twitter for those ISS times in Aurora. Get the magazine. Tell all your friends to get the magazine. Let's keep Astronomy Ireland the world's most popular astronomy society. So I'll just ask any of the volunteers listening in if I've left anything out. If not, we'll close things there for the evening. Over to you guys. They're very quiet. That sounds like everybody is happy. If you have any questions, contact the Society. We're always here to help. So good night, everybody, and thank you very much for joining us this evening. We'll see you all hopefully at the evening classes, October 3rd, and back here on Zoom on October the 9th.